Good morning, viewers at home. My name is Olatunde Michael. I am the CEO of Omega Bio-Edutainment. Today again, we are going to talk on the course which has to do with microbial genetics. Microbial genetics. Okay. Um, don't forget that the other time in the earlier videos, we actually look at cell biology, where we talk about prokaryotic and prokaryotic organisms. We were able to look at prokaryotic organism. An example of prokaryotic organism is that of a bacterial cell. And don't forget that in this course, we will be looking at the following. We want to first of all look at genetics. What do we mean by, if we talk about genetics, what do we actually mean? We also talk about a gene and its composition. We'll look at chromosomes. What do we mean? Chromosomes. Then we'll now look at the hereditary material. Then apart from that, don't forget we are talking about microbial, that is microorganisms, and the typical microorganism is that of a prokaryotic organism. So having look at that, we will try and also relate. Now, genetics, if we are talking about genetics is actually the science of the beginning. That is, from the word gene. Don't forget that gene is a segment. It's just a segment of DNA. Don't forget that in the earlier lectures, I told you that DNA is regarded as the alphabet. Is the alphabet of life. Is the alphabet of life. You also need to look at life from the angle of a cell. That is, so if you are talking about the alphabet of life, we are actually regarding DNA as the basis. DNA as the basis for which and on which life is actually made on. So talk about gene. Then now, if you now look at, let's look at a typical living cell. This is actually this one is a animal cell. This is an animal cell. So let's try and look at it like this. Okay. So okay. Now, chromosome is actually very, very prominent in animal cell or eukaryotic cell. So this is the plasma. This is the plasma membrane. This is a cytosol, or what you call part of it is a cytoplasm. Then don't forget that cyto is cell, then plasm is liquid. So that is the liquid parts of the cell is actually known as a cytosol or cytoplasm. So now this is a endoplasmic reticulum, which is concerned with the synthesis of a protein. Endoplasmic reticulum, and in this case it is a smooth endoplasmic reticulum because it has no ribosome that is attached. This is where we have the nuclear membrane. nuclear membrane. This is a nuclear membrane. So inside the nuclear membrane, we have nucleolus. Or nucleoplasm. Nucleolus or nucleoplasm. Don't forget that I told you that plasm is actually liquid. So the liquid inside the nucleus is a nucleoplasm. Now, inside the nucleolus now, that is where we now have our chromosome. This is where we have have chromosome inside the nucleus, and part of the genes are located, they are arranged serially on the chromosome. So this is where we have the gene on the chromosome. And don't forget that the gene is what I told you that gene is a segment of DNA. Now, don't forget the double helix structure of a DNA. So between here, a complete turn of DNA is actually regarded as a major groove. This is the major groove of a DNA. So in between here and here, we have between, depending on the type of DNA. Don't forget we have X DNA, Y DNA, nucleotides. Between this minor groove depend on the type of DNA, whether it is X, whether it is Y, or whether it is a Z chromosome. So between this and this is a complete turn. This is a complete turn of DNA. Now, the genes, the nucleotide bases are arranged. Then don't forget the base pairing rule, according to Chargaff rule, that says that in between C will always bond with what? With G. Then A will always C. So this is Chargaff rule, that is the number of C must be equal to G and the number of A must be equal to C. So all these are part of what we should know to be a kind of a relationship between gene, chromosome, and DNA. I've told you that a typical animal cell, which is an eukaryotic cell, part of the differences, you know, I told you in cell biology that the differences between eukaryotic organism and prokaryotic organism, one of the differences is just because in prokaryotic organism be regarded as organelles. So all these inside a big cell, that is the plasma membrane, inside a big cell we now have other smaller cells according to the theory of endosymbiosis. The theory of endosymbiosis. Don't forget that I told you that endo means internal, and symbiosis, that is mutualism. That is a kind of mutualistic relationship between small but complete organelles, which are called organs. That is, they are on their own. They are existing as independent. So in pro means primitive. Don't forget, pro actually means primitive, and karyotic, that is a carrion. Karyotic will come from carrion, and carrion actually means what? Either chromosomes, or what can be regarded as the nucleus. So that is telling us that a cell that is primitive, that has a incomplete or very primitive nuclear arrangement or specific nucleus is regarded as a prokaryotic, and that's why you have a typical example of bacterial cell. Now let's try and look at a typical bacterial cell, which is an example or representative of prokaryotic organisms. Okay.